Hey guys, welcome to our last topic in chapter 10. In this topic, we're going to talk about those assays and tools that we just talked about in 1 and 2, and how we can use these to even more explore different aspects of cell biology. And you can use those assays from before on standalone assays, or you can use them in these kind of techniques that we're going to talk about here. So this kind of rounds out our chapter here. And this is the last topic for this chapter and our last topic for this unit. So here's our topic objectives. This list is a little bit longer because it's more of an application section, but let me know if you have any questions about any of them. So first of all, let's talk about DNA sequencing. DNA sequencing is becoming more and more important. It was really important before, now it's becoming important again, because the more we understand about the human genome, the more we're understanding how specific it is and how it affects how we get diseases, how it affects how drugs work in our systems. And so if we can understand that, the area of personalized medicine can just be utilized even more heavily to help treat people better. So the big key is how do we sequence people's genomes quickly and inexpensively? And they're working on developing this even more. There's actually a competition to make sure that, or to help develop a human genome sequencing technique that's quick. But let's talk about the old school version. And this is through deoxy-DNA sequencing. And what we do in deoxy-DNA sequencing is we make these nucleotides, and these nucleotides have these, they're missing a chunk on the end of them. And what happens is they then can't bind to the next nucleotide, so it effectively stops the sequence from being built. What we do then is we will separate out sequences of the DNA, put one with a group of deoxy ATPs, deoxy TTPs, deoxy CTPs, and deoxy GTPs. And what they'll do is it'll read and then stop when it gets to, for instance, if we're in the deoxy ATP section, the first time it comes to an A, it's going to stop. So it'll put the A on and then that's the end. And then the next time it comes to an A, it'll stop. And that's just how we then sequence it. And you can see here that then we can just line up the bands, put the bands in order by size, and that allows us to then sequence the DNA. So we'll know exactly what the sequence is of that DNA originally. As you can see on the picture here, our sequence then clearly runs ATGTCAGG or AGTCCAG. And that's just how this deoxy sequencing works. So it's kind of cool, but it's cumbersome, especially if you're doing a long sequence of DNA. So now let's talk about how we use some of this information to construct new DNA molecules. So let's say that we want to make a new protein, or we want to modify a protein. So we need, or produce proteins that are rare. What we need to do is we need to modify that DNA. So we'll take that plasmid vector and we're going to insert the DNA with information that we want. And so what we do then is we can build this protein to be what we want. So let's say we want this total mutant hemoglobin protein. I insert the first gene, I insert the second gene, I insert the third gene, or of whatever I want, and however many genes I need into it, and it'll eventually then be folded into the protein I want. It can be a total mutant protein, it can be whatever it is that I want to do. Even if I wanted to just change out a subunit on a protein, I just want to change that subunit and see how that changes things. I can do that with this. So that's how this process of constructing new DNA molecules work. And we use restriction nucleases and ligase, just like we talked about before, to do this. We also can do that, as I said, to make proteins. And as I said, we use plasmids most often just because they're simple to use. One of the things that we can do when we're doing this, either the manipulating of the sequence or manipulating the proteins, we can add tags to it. So let's say that I want to be able to see this protein in the cell. I can go ahead and add in a GFP protein, or a GFP sequence, which stands for green fluorescent protein. It's a protein that'll glow green when it's being made, and when it's active in the cell. So I can easily see it in the cell because it's green. That's how I can do it here, is I can just add it on to the end of this sequence, and then the cell will produce this protein with the tag on it. Another option for making proteins in this case is that I can overexpress them. I can induce the cell to make a ton of this protein so that I can then have a ton of protein to work with if the cell, for instance, doesn't make a whole lot of it very quickly. So this is another tool. And we use those chromatography assays that we talked about in chapter 4 to help purify this process out, or purify this product out. So this kind of all circles back around to the circle of protein nucleic acid research. It's really a circle, or a cycle, I guess you could say. No matter if you start with the gene or the protein, you're going to end up circling back around to the other one. So if you start with protein, you can learn a lot about the proteins, but then you need to go back around to what the amino acid sequences are, how this works in the DNA database, look for it in the cells. If you start with the gene, you're going to want to know what that gene looks like as a protein, and you'll go from there. And either way, you can go and extrapolate these in both directions, but there is definitely an association. You can't do nucleic acid research without thinking about the proteins. You can't do the protein research without talking about the nucleic acids. As much as we pretend like they're separate, they really are one side of each of a coin. And I mentioned this before when we talked about manipulating the DNA, and this is adding in reporter genes. And as I said, one of the most common uses is green fluorescent protein. Now what this does is when it's triggered, makes the protein green. So you add it in, and then what you can do is you can check out what the regulatory sequences are. This helps us know how to turn on this gene. So if I wanted to know how to turn on the gene for lactase in a cell, I could insert my GFP where the lactase is, and then trigger different regulatory regions to see which one actually produces that GFP, and then I would know how I could turn on lactase. And so that's how this works here. And remember, it's not as simple necessarily as just turning on one gene in one cell equals all the answers. Sometimes you have to turn on multiple genes in one cell. Sometimes you have to, there's a different combination for different specialized cells. So it's really important to understand that. But using this reporter sequences is how we can find out what genes are working, when they're working, and what environment helps stimulate these genes. Now, another example of ways to track things within the cell or in other in situ situations is in situ hybridization. In situ means in place. This means that I've stained the cell, I've stained the chromosome, etc. One example that we use a lot is FISH, which is fluorescent in situ hybridization, which means I've taken my sequence of genes and I've hybridized them to the cell, and I've tracked now this item. And you can see a couple examples here on the slide. Makes things fluorescent. Whatever I want to look for, I can now see them in the cell. It's a different way without worrying about reporter genes or anything like that. And this is how we can see different regions on chromosomes, like you can see here. You can see different areas. It just depends on what we're looking for. This is also how sometimes we'll look for viral infections within cells. It's just a variety of ways. It's just using that tag to help us see that. That other hybridization technique we've talked about before, I'm not going to go into crazy detail here because we've talked about it before, is microarrays. And microarrays also use hybridization. So we take two samples of DNA, or in this case, the example is mRNA. We convert them, we color them either red or green, expose them to regions of complementary nucleic acid. So in this case, it would be an RNA. They bind, and then we look for which wells are green, which wells are red, and which wells are yellow. And if they're yellow, that's where green and red bound together. If they're red, that's only where the sample 1 bound. If they're green, that's where only sample 2 bound. And this tells us what genes are turned on and off. Now, if we want to find out gene functions beyond regulation and modifying the gene, we can do this through direct site-directed mutagenesis. So I can go in, if I know what my gene sequence is, go in and change one base. And then by changing that one base, I can see how that affects the entire gene. Now we use this a lot in genotypic and phenotypic work. Now, and it's very tedious because if we're just changing one base pair at a time, we change it, we see what happens. We change another one, we see what happens. Now you want to be smart, you don't just go through each one, because what do we know about codons? We know that codons don't always, changing a base does not always equal amino acid change. So we don't have to change every single one, but we do specific site-directed ones to cause a different amino acid to be there. And usually there's a lot of upfront work that goes into this, adjusting which amino acid we want, thinking about the different changes that we want. It's not just randomly changing things. But this allows us to find out what difference it is. So if I wanted to know what happens if I put an alanine in place of an asparagine, I can do that and then see, okay, this is what happens here. So this is how we can find out gene functions. Now, beyond the cell, well, a lot of this we do in the cell, but beyond the cell, we look at animal models and plant models. And in animal models, this is where we will create a couple different types of animal models. The first reason, one of the most common we talk about are knockouts. What this does is we get rid of the active gene. We silence it in some way, shape, or form. And so the animal no longer can make this gene. And so then we can either see what that results in, or we can then experiment on the animal and see how a drug or something interacts when that gene's knocked out. We can also do transgenic. This is where we could insert a mutated version of a gene. So like, for instance, if we know this gene seems to show up in patients with this disease. If we insert into this animal, will they have that disease? And then we can start to expand on that and understand how these genes are impacting the disease function. We can also insert more genes, and that can change things. Now, one of the keys to these kind of animal models is that they're very expensive, very time-consuming animals, and it's a really powerful tool, but it's also it's really expensive, really time-consuming. So it's not something you venture into lightly. We have to breed them very carefully to make sure that they maintain that knockout status or a transgenic status, whatever it is that we're after. And we have to watch them. And we almost always use recessive animals because of the fact that it'll show us gene changes a little bit faster. So it's just really important to understand that. So this is just a tool that we use, but it's a tool that gets used very sparingly just because it is so time-consuming and an expensive process. One way to do this easy in cells, like to make a knockout type thing or to control gene expression, is RNAi. And what we do here is in the cell, we insert this interference RNA. We talked about miRNAs and siRNAs earlier. In this situation, we insert these RNAis, and what it does is it artificially changes the gene expression. What it'll do is it'll knock down the mRNA so the gene can't be expressed in a process. Problem with this is that it doesn't get carried generation to generation. It's very short term, but it's a quick way to see if you even have a proof of concept before going into these more expensive processes. And as I said, just like making custom animal models, we can also do transgenic plants. It's a very similar process, except for in this case, we use a little bit different way to control that plant and grow up more copies, because obviously plants can be reproduced in different ways. And this is how the agriculture and food industries go about making insect-resistant crops and things like that, GMOs. And so this is just another way that this works. So this is the end of topic 3, this is the end of chapter 10, and this is the end of unit 2. Yay, you are officially halfway through the content for this class. As always, review those objectives and let me know if you have any questions at all, and we will talk about this more in class.